May 11. 100 datasets broke your alien life shortcut. Klenner's warning is simple: dead chemistry makes the same organic molecules too, so Yoffe's target is treating one clue like the whole case. For decades, missions hunted molecules one at a time. For decades, meteorites carried amino acids, so your alarm rang early. Synthetic space chemistry can make them too, which makes Klenner's trap worse. Before this week's paper, the hunt kept asking what molecule appeared. Klenner's point is life leaves ordering habits, not just ingredients. That sounds small until 100 datasets spread into separate chemical neighborhoods. Yoffe calls this a biosignature, meaning how much of each. In Yoffe's living samples, amino acids spread wider and more evenly. In the same study, fatty acids flip the pattern back. Mars, Europa, and Enceladus may read Yoffe's patterns next, and if that works, one clue becomes the whole pattern. This month, Yoffe's team fed chemistry tables through one simple test. They measured molecule variety and balance across every sample. Across 100 datasets, living amino acids spread wider than dead chemistry. So you are not chasing one molecule, you're reading the whole crowd. Meteorites can carry amino acids too, which is why this gets tricky. But life packs its amino acids with a strange statistical fairness. A dead reaction leaves a thin playlist, while biology fills the room. Now the second cascade flips the story, and that matters. For fatty acids, non-living chemistry looked broader than biological chemistry, meaning the pattern changes by molecule family, but it still separates processes. If you only hunt a magic molecule, you can fool yourself. If you read the organization, the false alarms start shrinking. That is why Mars ice, Europa spray, and Enceladus grains matter. They may not find life, but they may read its handwriting. On May 11, 100 datasets made the wrong shortcut look dangerous, because if you crown one molecule, chemistry can impersonate biology. The amino acid pattern showed life spreading its choices more evenly. Abiotic samples clustered hard, meaning nature kept repeating fewer options. Then fatty acids flipped it, and that reversal saved the method. So the test asks your sample for its whole chemical shape. That is the cascade one false headline can steer a mission. A rover has hours, not infinite chances to chase every molecule. Europa ice may carry signals, but radiation keeps sanding them down. Enceladus plumes offer seconds, so abundance becomes the practical clue. You are not looking for one trophy molecule anymore. You are looking for the pattern a living system leaves. In the control room, that pattern changes who speaks first. One exhausted scientist closes the file because life now looks organized. Not one molecule, the pattern across roughly 100 datasets breaks open. In living samples, amino acids spread wider and you see smoother balance. Rock chemistry makes amino acids bunch up like signals crowding one corner. Then the twist flips because fatty acids tell the opposite story. Cells favor certain fatty acids, so biology tightens that chemical spread. Untouched chemistry scatters fatty acids wider, meaning the molecule alone lies. So you need the whole table because one molecule can mislead. Europa may hand us messy organics, where ratios do the talking. Enceladus already throws ocean spray outward, and future instruments can count. This month, Nature Astronomy made alien hunting less romantic and more statistical. The UCR team avoids declaring life and changes the test. Instead of asking glycine, you ask whether the whole table behaves alive. That is the payoff. Alien life may show balance before beauty. Next, Mars hands you numbers, and one molecule was never enough.